what are the major events that occur during sliding filament theory? Now, as I told you earlier, number one, there will be release of calcium ions into sarcoplasm from the cisternae. Now, these calcium ions combine with the TNC. Calcium ions now they bind with the TNC molecule of troponin complex. Okay, they combine with the TNC molecule of troponin complex. The moment there is interaction of calcium with the troponin complex now, a kind of conformational change takes place within the troponin complex. The troponin complex and tropomyosin, both of them are pushed aside. Okay, a kind of distortion of the protein takes place by pushing troponin and tropomyosin aside. As a result, the binding sites of G-actin molecules are exposed. These are all G-actin molecules with their binding sites for myosin heads now. Now, these binding sites are concealed by like this troponin complex, as you learned here now. This is TNI, this is TNC, this is TNT, and this is tropomyosin. So, when calcium ions combined with the TNC molecule, the three of them are pushed aside. Tropomyosin is also withdrawn from the groove. Consequently, the binding sites of G-actin molecules get exposed. So, the same thing I will write here, students. So, what is the third event that occurs here now? First, there will be release of calcium ions into sarcoplasm. Second, calcium ions combined with the TNC. Third event, distortion of troponin occurs. Troponin complex occurs by exposing the binding sites of G-actin molecules. Okay, by exposing the binding sites of G-actin molecules. Now, there will be continuous movement or diffusion of calcium ions into sarcoplasm. Due to high concentration of calcium ions, there not only exposure of binding sites of G-actin, there will be activation of myosin heads. All the myosin heads which are parallel to thick filament when the muscle is at rest, they become perpendicular. All the myosin heads, when the muscle is at rest here now, all the myosin heads like this, they are all parallel like this to thick filament. Myosin heads are parallel to thick filament here now. But when the calcium ions are released into sarcoplasm here now, because of high concentration of calcium here now, the myosin heads become perpendicular to thick filament like this. The myosin heads become perpendicular to thick filament. All the myosin heads become perpendicular to thick filament. When they become perpendicular, they come in contact with the G-actin molecules. They come in contact with, this is thin filament here now, this is Z line. So, they come in contact with G-actin molecules. The entire thin filament is made up of G-actin here now. So, the myosin heads come in contact with G-actin molecules to form actomyosin complex. Once actomyosin complex is formed, there will be activation of ATPase. So, what is an activation of myosin heads to form actomyosin complex? Actomyosin complex. Now, when actomyosin complex is formed, ATPase is activated. ATPase enzyme is activated. Now, the activated ATPase splits the ATP into ADP plus IP. Now, where is this ATP present? ADP will be there in the sarcoplasm here now. Once the myosin head comes in contact with the actin, each actin ATP molecule goes and attaches to the myosin head here now, interacts with the myosin head. And now the activated ATPase splits the ATP into ADP and IP, adenosine diphosphate inorganic phosphate here now. When there is a hydrolysis of ATP into ADP and IP, first IP is released into sarcoplasm. With the loss of IP, the myosin head no longer remains perpendicular, it gets tilted like this. It gets tilted like this. Okay, each molecule gets tilted like this. The tilt of myosin head is called power stroke. Now, while tilting itself, it pulls the thin filament towards the head zone here. Okay, as long as it is straight, it will be in contact with the G-actin. When the ATP is hydrolyzed into ADP and IP, due to loss of IP, the thin filament gets tilted here. The tilt of or the swiveling of myosin head is called power stroke. While tilting itself here now, it pushes that thin filament towards the head zone. Then with the loss of ADP, the entire myosin head once again becomes parallel like this. When it becomes parallel, one more ATP goes and attaches to the myosin head. Calcium ion concentration is still high in the sarcoplasm. In the presence of abundance of calcium here now, ATP once again goes and attaches to each myosin head. When there is attachment of ATP, once again myosin head becomes perpendicular, coming in contact with subsequent G-actin molecule, because the first G-actin has already been pushed inside here now, so it cannot come in contact with the same G-actin. Each myosin head comes in contact with the subsequent G-actin molecule to form actomyosin complex. Once again, when actomyosin complex is formed, ATP is activated, splitting the ATP into ADP and IP. Then there will be loss of IP. With the loss of IP, there will be swiveling movement of the myosin head, that is tilt of myosin head, which is called power stroke. While tilting itself, it further pulls the thin filament towards head zone. Then with the loss of ADP, it completely retracts itself from the G-actin and becomes parallel. Once again, because of attachment of ATP, the myosin head once again becomes perpendicular. So remember students, for the molecule to become straight and for the myosin head to become straight, myosin head to interact with the actin, ATP is required here now. And when there is hydrolysis of ATP, energy that is liberated is utilized for withdrawal of the myosin head. So ATP is required for the attachment of myosin head with the G-actin molecule. Energy that is liberated from the ATP is utilized for withdrawal of the myosin head from the G-actin molecule. When the IP is lost, inorganic phosphate is lost, there will be a power stroke. When ADP is lost here now, entire myosin head withdraws itself from the G-actin and becomes parallel to the thick filament here now. This attachment and the detachment of myosin heads here now is called ratchet movement. So the ratchet movements are responsible for shortening of the muscle fiber. Now dear students, remember again EAMCET, Comet K and other exam point of view, you must remember these points now. The changes that occur during contraction process. During contraction process, these thin filaments are pulled towards the head zone by means of myosin filaments. When thin filaments are pulled towards the head zone, along with thin filaments, Z lines are also pulled towards the head zone. As the Z lines start moving towards head zone here now, head zone goes on decreasing in size. At a point of time, entire head zone disappears. Entire A band appears dark. So during contraction, head zone decreases in its size, head zone completely disappears, entire A band appears dark, and length of sarcomere decreases because of inward movement of Z lines towards the head zone here now. Now all these changes occur as long as ATP and sarcoplasm, calcium ions in sarcoplasm are available abundantly here now. When ATP is exhausted or when there is no stimulation from the motor end plate, all the calcium ions are once again reabsorbed by the cisternae of sarcoplasmic reticulum. In the absence of calcium ions, once again troponin complex restores its normal position, concealing the binding sites of G-actin here now. Tropomyosin also extends into the groove of two effectin strands. Troponin assumes its normal position here now, blocking the binding sites of G-actin. Consequently, the myosin heads will no longer interact with the G-actin molecules. All the myosin heads withdraw themselves from the G-actin molecules and become parallel to thick filament here now. When the myosin heads withdraw themselves now, once again the thin filaments start moving away from each other along with their Z line. They start moving backwards towards their normal position along with their Z lines. So relaxation occurs maybe because of loss of ATP, number one, or because of lack of stimulation, number two, Or number 3, loss of calcium ions. When there is lack of stimulation, all the calcium ions are reabsorbed by cisternae. When there is lack of ATP also, muscle cannot contract further. In such case, all the myosin heads withdraw themselves from the actin filaments and become parallel to thick filament. The thin filaments move away from each other along with the Z lines. Head zone reappears. Entire A band once again appears dark only on lateral sides and light in the middle. So such changes occur during relaxation process. So remember students, the events that occur during contraction, events that occur during relaxation are important. Your EAMCET, COMET K, AFMC and JIPMER point of view. Now we shall throw some light about the biochemical events that occur during contraction process. Now biochemical events that occur during contraction. First, there will be utilization of sarcoplasmic ATP. The ATP present in the sarcoplasm is first used up by the muscle for contraction process. When the muscle is made to contract vigorously, the sarcoplasmic ATP gets exhausted within no time. When sarcoplasmic ATP is exhausted, muscle derives ATP from ADP. Energy is derived from ADP, adenosine diphosphate. How does adenosine diphosphate generate ATP? Look at these students. ADP, in the presence of enzyme myokinase, splits into AMP and IP. In the presence of an enzyme myokinase, splits into AMP and IP. Now that IP, inorganic phosphate, combines with the remaining ADP molecules to form ATP. Suppose there are 100 ADP molecules in sarcoplasm, maybe 50 get catabolized to form AMP and IP. The IP will combine with the remaining 50 ADP molecules to form ATP. Now this ATP enables the muscle to contract for some more time. Now during strenuous contractions, even that particular ADP also gets exhausted. Now when ADP is exhausted, muscle derives energy from a phosphogen called creatine phosphate. Muscle derives energy from a phosphogen called creatine phosphate. Remember students, in all vertebrates including human being the phosphogen of the muscle is creatine phosphate, whereas in invertebrates it is phosphoarginine. Okay, in vertebrates it is called creatine phosphate or phosphocreatine, whereas in invertebrates it is phosphoarginine. Whereas in hemichordata, then chordates, hemichordata, echinodermata and chordata. I repeat once again, hemichordata, echinodermata and chordata. In all three of them, okay, the energy rich protein called creatine phosphate is present, and especially in the hemichordates, in echinoderms and hemichordates, both CP and PA are present. Okay, both creatine phosphate and phosphoarginine both are present in echinoderms and hemichordates. That's why they are considered as connecting link between chordata and non-chordata. Whereas in all chordates, all vertebrates, creatine phosphate is the phosphogen. In all invertebrates, phosphoarginine is the energy rich protein, whereas in hemichordates and echinoderms, both creatine phosphate and creatine arginine, that is phosphoarginine, are present as energy rich proteins. So when the ADP is exhausted, muscle derives energy from a phosphogen called creatine phosphate. How does creatine phosphate generate NAP? Look at these students. Creatine phosphate, in the presence of an enzyme creatine kinase, splits into creatine plus inorganic phosphate. In the presence of an enzyme creatine kinase, it splits into creatine plus inorganic phosphate. That inorganic phosphate combines with the ADP molecules to form ATP. Now this ATP sustains the contraction of the muscles for some more period of time, enables the muscle to contract for certain period of time, When the muscle is made to contract vigorously, even the CP gets exhausted, When the CP is completely exhausted, muscle synthesizes ATP by anaerobic glycolysis, Instead of resorting to aerobic method, muscle prefers to synthesize ATP by anaerobic glycolysis. Now during anaerobic glycolysis, glucose or glycogen is broken down to form pyruvic acid. This pyruvic acid is broken down to form either ethyl alcohol or lactic acid by releasing certain amount of ATP. So here glucose is broken down into pyruvic acid through glycolysis. Under normal conditions, pyruvic acid enters Krebs cycle to synthesize ATP, which is referred to as aerobic respiration. But during strenuous contractions, when the CP, then ADP, then ATP, when they are exhausted, muscle makes a fast buck of ATP through anaerobic glycolysis rather than aerobic method. Now during this anaerobic glycolysis, pyruvic acid is broken down to form ethyl alcohol or lactic acid. Yes, students, ethyl alcohol formation takes place in bacteria, fungi and all other certain parasites, whereas in human being and vertebrates, there will be formation of lactic acid along with the ATP. Now this ATP enables the muscle to contract for a certain period of time. Now when the entire pyruvic acid is converted into lactic acid, even ATP is also completely exhausted. Muscle now stops responding to the external stimulus. That non-responsive phase of the muscle due to exhaustion of all the energy resources is known as muscular fatigue. What is that known as? Muscular fatigue. So muscular fatigue crops up due to depletion of energy resources within the sarcoplasm like ATP, ADP, CP and pyruvic acid. The another reason is accumulation of lactic acid here. As long as lactic acid is present in the muscle cannot respond to external stimulus. It remains in a non-responsive phase known as muscular fatigue. Now skeletal muscle overcomes fatigue through a phenomena called oxygen debt and Cori cycle. Now students, we shall talk about now oxygen debt and Cori cycle, which are the most important events that occur in the muscle, and the most important events for your EAMCET, Comet K, JIPMAR, AFMC, so on and so forth. Now oxygen debt. What is oxygen debt? A person while running, if he consumes say about 4 liters of oxygen per minute, when he is about to stop an event, he may consume more amount of oxygen. He may consume nearly 6 to 7 liters of oxygen when the person is about to stop that strenuous physical activity by panting, wheezing and gasping. That additional amount of O2 which is inhaled by the lungs towards the fag end of a muscular strenuous activity is utilized for combustion of lactic acid. So the additional amount of O2 inhaled by a person which is utilized to burn lactic acid, to oxidize lactic acid in the muscle is known as oxygen debt. So while running or while performing a strenuous physical activity, the amount of oxygen consumed may be less, but when the person is about to stop the event he consumes double the amount of oxygen. That additional amount of O2 which is inhaled, by a person to burn about 1/5 of lactic acid to convert into ATP plus CO2 is called oxygen debt. That is, the amount of oxygen inhaled by a person is not sufficient to burn entire lactic acid into ATP. 1/5 of lactic acid gets burnt in the presence of additional amount of O2 to form ATP and CO2 Now, in the presence of that additional, in the presence of this ATP remaining amount of lactic acid, that is 4/5 of lactic acid 4/5 of lactic acid is converted into glucose in liver in the presence of various enzymes, and this process is called Cori cycle. So I shall repeat once again. Additional amount of O2 inhaled by a person utilized to burn 1/5 of lactic acid to form ATP and CO2 is called oxygen debt. In the presence of that ATP and the presence of various enzymes here now, remaining amount of that is 4/5 of lactic acid here now, 4/5 of lactic acid, okay, 4/5 of lactic acid is converted into glucose in liver through a process called Cori cycle here now. When the glucose is formed, now the glucose gets converted into glycogen here now. From liver, the glycogen may enter muscle. In the muscle, once again, it is oxidized under normal conditions through aerobic method to synthesize as many as 38 molecules of ATP here now. When ATP is utilized, once again, the energy-rich compounds which have been utilized during contraction are once again replenished here now. So remember, students, two events which occur during contraction, that is oxygen debt, additional amount of O2, which is inhaled by a person when the person is about to stop a strenuous physical activity, which is utilized to burn only 1/5 of lactic acid to ATP is called oxygen debt. And Cori cycle is conversion of remaining amount of lactic acid, maybe 4/5 of lactic acid or 80% of lactic acid to glucose in liver is called Cori cycle here now. Glucose, once it is formed, glucose is converted into glycogen. From liver, glycogen enters into muscle. In muscle, once again it is burnt through normal process to form lactic acid. Once again lactic acid enters liver to form glucose, like the cycle goes on here now. So the process of conversion of lactic acid into glucose or glycogen is called Cori cycle. The process of combustion of 1/5 of lactic acid or 20% of lactic acid to yield ATP in the presence of additional amount of O2 is called oxygen debt here now. So a person overcomes fatigue through oxygen debt and Cori cycle here now. Then once sufficient amount of ATP is formed here now, the muscle gets ready to contract further upon stimulation here now. So these are the major events that occur during contraction. Now the students here now, the most important concepts in the musculoskeletal system we shall talk about now, and these are all important. You have AFMC, JIPMER, AIMS, then COMET-K, all exam point of view. Okay, the most important concepts here that you come across in the muscle physiology. Starting with rigor mortis. Now what is rigor mortis here now? The stiffening of the muscles soon after death of a person. Stiffening of muscles soon after death of a person can be called rigor mortis here now. Then when the person is about to die here now, all the cross bridges of the myosin heads will be interacted with the actin filaments here now. For the detachment of that, ATP is required here now, or hydrolysis of ATP is required. Hydrolysis of ATP takes place only in the presence of an enzyme called ATPase enzyme. When the person is about to die, all the enzymes get denatured. Consequently, the myosin heads cannot withdraw themselves from the actin filaments. They remain attached to the actin filaments you know. Consequently, most of the skeletal muscles remain in contractile form. Because of the contractile nature of the skeletal muscles, the entire body becomes stiff. The stiffening of the muscle fibers soon after death of a person can be called rigor mortis. In the rigor mortis stage, the person may remain nearly for 4 to 5 hours. And the rigor mortis stage here now, the person is said to be fresh. The bacteria, then different kinds of pathogens present in the atmosphere dare not enter the human body because of high level of lactic acid. High level of lactic acid within the body. Because you learnt already here now, mostly during the fatigue stage here now, the entire muscle gets deposited with the lactic acid. Because of large amount of lactic acid accumulation in the muscle here now, because of high acidic nature and low pH here now, the pathogens present in the air cannot enter into the dead body. As a result, muscle remains fresh. So the freshening of the muscle fiber or fresh nature of the muscle fibers and stiffness of the muscle fibers soon after death can be called rigor mortis you know. Then when the person is in the rigor mortis stage here now, slowly the lactic acid starts decomposing here now. As it decomposes, then the bacteria, then fungi and all other pathogens present in the air, they enter on a warfoot manner into the dead body and start decomposing the muscles also. Because of decomposition of the tissues and muscles now, body becomes flabby and body starts emanating foul smell. But as long as it is in the rigor mortis stage here now, body remains fresh because of high content of lactic acid. As the lactic acid starts decomposing here now, there will be invasion of the pathogens into the human body, dead body here now, bring about autolysis. Because of autolysis, body starts emanating foul smell here now. So that is called rigor mortis stage. Then next, students, we shall talk about red muscle fibers and white muscle fibers. Differences between these two muscle fibers, most important events, which can be asked in Comet K, AFMC and all that. Now we shall do that, Now, red muscle fibers and white muscle fibers, questions were asked in the previous exams based on even red and white muscle fibers also. So, you should be aware of the important characteristics of the red and white muscle fibers. Red muscle fibers, dear students, are also called tonic muscle fibers. Tonic muscle fibers or slow tonic muscle fibers or slow twitched muscle fibers. These are called fast twitched muscle fibers. They are called the tonic muscle fibers or slow twitched muscle fibers. They are called fast twitched muscle fibers. Now, the red muscle fibers are thin. These are thick. Okay. These are white muscle fibers are bulky, whereas red muscle fibers are thin. Now, the red muscle fibers, they have abundance of mitochondria. Abundance of mitochondria. Mitochondria are sparse in this. They are sparsely developed in white muscle fibers. Now, in red muscle fibers, the sarcoplasmic reticulum is poorly developed, whereas SR is well developed in white muscle fibers. SR is well developed, sarcoplasmic reticulum is well developed in white muscle fibers. Then red muscle fibers have got less amount of glycogen. They have abundant amount of glycogen. Abundance of glycogen is noticed. Red muscle fibers, they are red in color due to deposition of a respiratory pigment called myoglobin. The sarcoplasm is deposited with respiratory pigment called myoglobin, large amount. That's why they appear red here now. Here myoglobin content is less or absent. Myoglobin is less or absent. That's why you call them white muscle fibers. Now these red muscle fibers undergo slow and sustained contractions. Slow and sustained contractions. Whereas the white muscle fibers undergo vigorous contractions. They undergo slow and sustained. Contractions are very slow and sustained, whereas they undergo vigorous contractions. So the deposition of myoglobin is noticed. Here myoglobin is less or absent. The slow and sustained contractions, whereas in case of this, contractions are fast and vigorous. Contractions are fast and vigorous. Now these are innervated by thin nerve fibers. These are innervated by thick and stout nerve fibers. Thick and stout nerve fibers, because giant axons always conduct impulse very fast, so they require giant axons, whereas they are all under control of thin and small nerve fibers. Now they synthesize ATP mostly by aerobic method. ATP is synthesized by aerobic method, whereas here they synthesize ATP by anaerobic method. Okay, they undergo contractions, very fast and vigorous contractions, so they require ATP quickly. They cannot rely upon the aerobic method because the aerobic process is a slow process, takes a lot of time to synthesize ATP. Till then the muscle cannot wait. That's why it mostly makes a fast buck of ATP through anaerobic method, whereas this synthesizes ATP slowly by aerobic method. Hence the contractions are slow in this, contractions are fast in this, They are under the innervation of thin nerve fibers, they are under the innervation of thick nerve fibers, Here they don't require mitochondria because respiration is anaerobic respiration, which can take place inside sarcoplasmic reticulum or in cytoplasm. That's why sarcoplasmic reticulum is well developed here, whereas here mitochondria are well developed here now. Okay, they've got abundance of, one more point you can write here now. Here, amount of oxygen present in the muscle, O2 is very high, here it is low. Because of abundance of oxygen here it prefers to synthesize ATP by aerobic method. Because of lack of oxygen here it synthesizes ATP by anaerobic method. So ATP synthesis by anaerobic method here Anaerobic method. Here ATP synthesis takes place by aerobic method. Since the contractions are very slow, they have abundance of oxygen, abundance of glucose here they do not undergo fatigue. Red muscle fibers do not undergo fatigue. Whereas they undergo fatigue quickly. They undergo fatigue quickly. White muscle fibers undergo fatigue quickly here now. Whether you differentiate between thick and thin muscle fibers here now, then what are the examples now? Here the muscles that sustain, muscles of the back that sustain your body posture that sustain our body posture or red muscle fibers. You are able to stand here and able to sit here because of contraction of the muscles of the back, they contract very slowly and remain in the contractile phase for a long period and thereby enable you to maintain body posture, So they are all red muscle fibers. Whereas the muscle fibers of eyelids, eyelid muscle fibers are the white muscle fiber because contractions are very fast and rapid here now. So eyelid muscle fibers are white muscle fibers. The muscles present in the back are red muscle fibers here now. But dear students, remember, depending upon the exercise undertaken by a person here now, a red can transform even into white or vice versa here now. Like people, those who take part in the 200 meter dash, 300 meter dash, a 400 meter dash, they have to run slowly where they need red muscle fibers, But people, those who take in athletic events, other like shot put, javelin throw, so on and so forth, high jump, long jump and all that, they have got white muscle fibers. So depending upon the exercise also what you undergo here now, there will be transformation of white into red and vice versa here now. So people, those who take part in athletic events like javelin throw or shot put or high jump or low jump or swimming here now, they have got more number of white muscle fibers here now. Then people, those who take part in marathon race, 400 meter dash, so on and so forth, they have got red muscle fibers, In the same way, birds you take, for instance, sparrows, they fly very fast only for short distances. Fly very fast only for short distances, So they have got white muscle fibers, They fly only for short distances and very fast they fly here now. So they got white muscle fibers, Whereas kites and vultures, they fly for a long period of time here now. Sustained flying habitat is noticed in them. So they require red muscle fibers, Same way the fish here now, like mackerels, tunas and all that, while cruising, that is while moving slowly, they make use of red muscle fibers here now. And while running very fast, they make use of white muscle fibers, Okay. Like that, the muscles, depending upon the conditions of the body, they can become either red or white, Yeah, some more concepts, students, we shall talk about now. All or none law. What is all or none law is also called Bowditch law. This all or none law is also called Bowditch law. Stimulus is given, either muscle contracts or does not contract at all. Usually the cardiac muscle responds to the threshold stimulus. Whereas skeletal muscles respond even to low threshold or high threshold stimuli also. What is threshold stimulus? The minimum intensity of stimulus that evokes maximum response in a muscle fiber can be called threshold stimulus. So cardiac muscle fiber always responds to threshold stimulus. If the intensity of the stimulus is sufficient enough, then it contracts, otherwise it does not contract at all. In case of skeletal muscle fiber, even if it is low threshold, the muscle shows response, because in case of skeletal muscle, the entire muscle does not obey all or none law. It is only that a bundle of muscle fiber obeys all or none law. Okay, you learnt already earlier. That is, a bundle of muscle fibers along with the motor neuron can be called motor unit. So in case of skeletal muscles, only motor units obey all or none law, whereas in cardiac muscle, the entire cardiac muscle obeys all or none law. So what is all or none law? When a stimulus is given, if it is threshold, muscle contracts. If it is non-threshold, muscle does not contract at all. Okay, it does not show any response. Called Bowditch law or all or none law. The next you have another concept called twitch. Now what is twitch? A simple contraction that is elicited in the muscle fiber by a threshold stimulus can be called twitch. So a simple contraction elicited in the muscle fiber by a threshold stimulus. By a threshold stimulus can be called twitch. A simple contraction elicited in the muscle fiber by a threshold stimulus can be called twitch. Now the twitch consists of three phases. Here you have latent period. Second phase is called contraction phase. And the third phase is relaxation phase. So all these three phases can be put together called twitch. Latent period is the duration between point of stimulation and commencement of contraction. During this, you find the transmission of impulse from the motor end plate to sarcolemma, depolarization of sarcolemma, then transmission of stimulus from the sarcolemma to the myofibrils through triad system, then release of calcium ions, then conformational change in the troponin complex, then binding of myosin heads with the actin filaments. So all those changes occur during latent period. Then latent period lasts for about 0.01 second. It lasts for about 0.01 second. Contraction phase involves power strokes and ratchet movements. It involves power strokes and ratchet movements. Where there will be pulling of thin filament towards head zone. Head zone goes on decreasing in its size. Then finally disappears. Z lines are also pulled closer to the A band. The length of sarcomere decreases. So all those changes occur during contraction phase. Contraction phase lasts for about 0.04 seconds. 0.04 seconds. Okay. The next is relaxation phase. Relaxation phase is the phase during which all the calcium ions are reabsorbed by the cisternae. Myosin heads withdraw themselves from the thin filaments. Thin filaments start moving away from each other. Head zone reappears. Length of sarcomere increases. A band appears dark only on the lateral sides, light in the middle. So all such changes occur during relaxation phase. It may last for about say 0.04 second to 0.05 seconds. So the entire duration of twitch is about say 1 second. Duration of switch is about 1 second. 0.01 plus 0.04 plus 0.05. altogether it amounts to 0.1 second. So a twitch is a simple contraction elicited in the muscle fiber by a threshold stimulus can be called twitch. Next we have another concept called summation. What is summation here now? A muscle is given a threshold stimulus. A muscle is given a threshold stimulus. It starts contracting. Before it could relax, before the muscle could relax, before the muscle could gain relaxation phase, if another stronger stimulus is given, it undergoes further contraction whose tension is much higher than the previous one. I shall repeat once again. While the muscle is in the contractile phase, before it could relax, if another stimulus is given, the second stimulus evokes much higher contraction than the first one. The second stimulus elicits much higher contraction than the first one. So that phenomena of a stronger contraction being evoked in the muscle fiber by giving another stimulus can be called summation. There will be summing up of the two waves of contraction, which is called summation. So a muscle, while it is in the contractile phase, before it could attain relaxation phase, if another stronger threshold stimulus is given, the contraction elicited second time will be much higher than that of the first time, which can be called summation. Okay. Then next we have called tetanus. Tetanus is a stimulus is given, a threshold stimulus is given here now. When the threshold stimulus is given, it exhibits, as you learned here now, it undergoes a twitch here now involving latent phase, then the contraction phase, relaxation phase. After completion of the relaxation phase, if another stimulus, series of stimuli are given without much delay, several muscle fibers start contracting vigorously one after the other. Okay, several muscle fibers, or you can say several bundles of muscle fibers start contracting vigorously one after the other. The sustained contractions of the muscles by giving repeated stimuli at shorter intervals is known as tetanus. Sustained contraction of the muscle by giving several stimuli at shorter intervals one after the other is called twitch. Is called tetanus. So when a single threshold stimulus is given, muscle starts undergoing twitch. Okay. Then after relaxation, if another stimulus is given, then some more muscle fibers start contracting. For instance, you take a muscle body, either bicep muscle or tricep muscle, it may be made up of say around 10 to 15 bundles of muscle fibers, 10 to 15 fascicles. When a stimulus is given, first threshold is given, 1 or 2 fascicles will contract. Okay. Then when another stimulus is given, some more fascicles start contracting. That is, these 2 fascicles may relax. The other fascicles which are at rest, even they start contracting. So if you want to make all the fascicles contract at a time, you should keep on giving several stimuli at shorter intervals, okay, without any gap, so that you find sustained contractions in the skeletal muscle, which can be called twitch. Now that can be represented in the graph in the form of sustained contractions by giving several stimuli without much gap one after the other, making the muscle fibers contract vigorously is known as twitch. Okay, because twitch always involves contraction of several bundles of muscle fibers. Here, as I made a mention earlier, in case of skeletal muscle, it is a bundle that is obese all or none, which is also called what is that a bundle of muscle fibers, and motor neuron can be called motor unit. So a motor unit obeys all or none. When many stimuli are given several motor units take part in contraction. As a result high rate of contraction is noticed in that particular muscle here. High tension is noticed in that particular muscle here. So that sustained contractions of the muscles by giving repeated stimuli without much gap one after the other, involving several bundles of muscle fibers, is called tetanus. Then you have next one, trepi. Trepi is when several stimuli are given the rate of contraction goes on increasing with a high magnitude which can be called trepi. In the form of a staircase phenomena when the stimuli are given at shorter intervals repeatedly, several bundles of muscle fibers start contracting vigorously, showing high rate of magnitude of contraction which can be called trepi. Okay. It is also called staircase phenomena. The next we got another concept called isometric and isotonic contractions. When a stimulus is given to a muscle here now, the muscle contraction that is evoked in the muscle can be called tension. Okay, the contraction that is evoked in the muscle fiber can be called tension here now, and pressure exerted on a muscle by a load can be called force. The pressure exerted on a muscle by a load called force here now. Contraction which is evoked within the muscle fiber can be called tension here now. So during contraction, if length of the muscle remains same, but tension of the muscle increases, you call it isometric contraction. So isometric contraction here now, length remains same, length of the muscle remains same, but tension of the muscle is more. What is tension? That contraction that is evoked in the muscle here now, okay, is called tension. Okay, the tension of the muscle is more, but length of the muscle remains same. Length does not change, but tension of the muscle increases because of formation of more cross bridges and all. Tension may increase here now, but length remains same. Such contractions are called isometric contractions. Like suppose you lift a book from a table here now. Okay, the contraction that takes place, okay, in the muscle can be called isometric contraction here now. Okay, length of the muscle remains same, whereas tension of the muscle is more, called isometric contraction. Now isotonic contraction here now, tonicity of the muscle, that is tension, tonicity or tension remains same, but length of the muscle changes. Length of the muscle changes. It is called isotonic contraction. Tonicity or force of the muscle, tonicity or tension of the muscle remains same, but length of the muscle changes. You call it isotonic contraction. If length remains same, you call it isometric contraction. Like for instance, you push your hand against the wall, push the hand against the wall. The contraction that is evoked in the muscle is called isometric contraction. When you push your hand against the wall or against a mobile item, work is not done, whereas length remains same, but tension of the muscle is more here now. Okay, when you try to lift a small book here now, whose load is much lesser than the force that is exerted in the muscle, then it is called isotonic contraction. So isotonic contraction, tension remains same, but length of the muscle changes, whereas isometric contraction, length remains same, but tension of the muscle is more. So we differentiate between two kinds of contractions here now. Isometric contraction, that is when you lift heavy load here now, whose resistance offered is much higher than your muscle capacity here now, then it is called isometric contraction, where length remains same, but tension is more. Okay, then isotonic contraction here now, the tension increases, but the length remains same here now. When you lift a small load here now, the contraction that is noticed is called isotonic contraction. When you try to lift a heavy load here now, the contraction that is noticed is called isometric contraction. So these two kinds of contractions come across in the muscle fiber, isometric and isotonic contractions. Now all these characteristics are studied by means of kymograph, by means of an instrument called kymograph. All these characteristics in the muscle can be studied by using an equipment called kymograph. And this can be asked in the NEET or any other exam. Remember students, kymograph is an instrument which is used to record various physical events of the muscle here now.